So I've heard much about Lethbridge over the many years from Bruce, and so I'm really pleased to have the opportunity to be here. And I'm particularly honored to be asked to give the Brenda Milner Lecture because of the fact that I've greatly admired her work over many, many decades. I think she's over 100 years old now. So there are probably only a few people in the room here who could remember what the field of the hippocampology was like before Brenda Milner. Put up your hand if you can remember. Nobody? Okay, a few people are willing to admit it. Okay. No. So I break up the study of the hippocampus into two periods. There's BB and AB, before Brenda and after Brenda, because she changed the way we think. I mean, our sort of the assumptions, the conceptual framework for how to think about how the hippocampus is organized and how it helps the rest of the brain, the cortex. And really, when you make a change like that, it really, the whole field, I mean, has impact that lasts for many, many decades. And that's a fairly rare occurrence. Usually science is incremental, where you add another brick to the wall and make progress that way. And so it's a very special person that has that kind of an impact. So I want to take you back as a kind of an introduction to a book chapter that, as you can see, Pat Churchland, Ramachandran, and I wrote back in 1994. So this was for a very pretentious book about large-scale neuronal theories of the brain. And, you know, the reality is that we don't have one, and we still don't have one. We have lots of insights and models and you know, hypotheses and so forth, but it's really very, very difficult to think about theories on the same scale that physics has theories. We just don't have a theory like quantum mechanics for how the brain works. Maybe we shouldn't, but I'm just saying what the state of the art was back then. Now, what we did is a little bit naughty, because we went through the literature on vision, and we tried to ask the question of whether or not the fundamental assumptions that everybody in the field of vision, both biological vision and computer vision, whether their assumptions held any water from the data. And as you can see, this title here is from Pat Churchland, who, by the way, is a Canadian, a colleague of mine for many years at UCSD in the philosophy department, A Critique of Pure Vision. And it has to do with the fact that in vision, there's this modularity hypothesis, that it's kind of a module in the brain, or in computer vision, it's a program which is separate from the rest of the rest of the brain. In the case of computer vision, it's a module that feeds into the decision module and the planning module and the motor module, right? It's kind of sitting there pre-processing all the sensory information. And here are a couple of quotes from researchers who have had a lot of impact. David Marr. How many people here remember David Marr? Okay, so he still has an impact. And this is his book, Vision. Its purpose is to build a description of the shapes and positions of things from an image, which seems very reasonable. I mean, after all, you look out and you see things. You see objects. You see a scene. More detail, this Alamonis and Rosenfeld in 91, from computer vision, regarding the central goal of vision as scene recovery makes sense. If we are able to create, using vision, an accurate representation of the three-dimensional world and its properties, then using this information, we can perform any visual task. So here's a very explicit goal. We want to write a program, and it's going to have inside the program, it's going to be a miniature model of what's out there. So we have to convert two-dimensional image into a three-dimensional internal model. And once we have that, then we can send the coordinates off to the motor system. So if I wanted to reach out for something, I know how far to go and what to do. And that's what they've been trying to do for years and years and years. And it turned out to be much more difficult than anybody imagined. So here's an example of something that we came across. It's called change blindness. Now, it's blinking once every second, and when it does, some object and it disappears. And I want you to figure out, where is that object? Put up your hand when you see it. I'd say about 10%. How did you miss it? I mean, it's like your nose, you know, how could you miss something that big? And here's what we now think. We think that you don't create a complete internal model of the scene around you, but rather just parts of it that you are attending at any given moment. And if you're not attending to that little patch, you're not going to see it change. Now, here's something even more interesting, okay? Now that you see it, can you not see it? It's impossible to ignore, right? So that tells you something else about vision, which is that once you've attended to something, it changes your brain. Your brain isn't the same anymore. Okay, so everybody has the idea. Now I'm going to give you another similar scene, and it's going to be an intelligence test. And the first person that puts up their hand and sees the difference, it's going to be a little more subtle, is going to win the prize. Okay, is everybody ready? On your mark, get set, go. Bump, 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 bump. One, okay, we have a winner. What is it? A VQ, vision quotient. Okay, so this was just one of many phenomena that are out there that are telling us that our intuition about our visual system is wrong. That is to say, it feels as if we can see everything, but no, it's an illusion that you see everything. No, you don't see everything. We have a fovea, and you see what you're looking at or what you're attending to, but not everything at the same time. But it turns out there's a huge literature out there with little things in it which didn't make any sense. Like, for example, there were papers out there saying that we have detected auditory signals in the visual cortex. How did they get there? Well, this paper is ignored. You know, what the heck? Maybe it was a stray artifact. Or even more dramatic, in the LGN, the lateral geniculate nucleus that feeds primary visual cortex, motor signals have been detected. What are motor signals doing in the visual system? Wow, I mean, this really is puzzling. So here's another example. This is from Paul Glimcher's lab, and it's an incredibly simple idea. It takes weeks and months to train monkeys to do a task, even a simple task. And the way they do that is by giving the monkey a drop of juice every time it gets the right answer, and no juice if it gets the wrong answer. And everybody, since time immemorial, has been using one drop of juice. Now, Paul Glimcher had the idea, what if we use 2 drops of juice? Will that change anything? So here's the task. The task actually has 2 blocks. In both blocks, it's exactly the same task. You fixate, a light goes on either to the right or left, and you saccade to the light. And if you hit the light within a certain window, you get your drop of juice. I mean, what could be simpler? The difference between the 2 blocks is that in the first block, the red block, you get twice the amount of juice than to the left, and vice versa for the second block. And the monkey knows that. He knows what block he's in, okay? And then they may go on for dozens of trials at each block. Now, this is a recording from a neuron in the parietal cortex that gets input from the visual stream, the dorsal visual stream that's important for localizing things in space, and also has motor signals that are important for issuing commands, for example, for eye movements. So here's when the target appears, and first let's focus on block 2. Here it's getting half when it moves to the right. So these are all rightward motions, and this is a raster. Each of those little ticks is a spike. So there's about a couple dozen trials here. Here's the post-stimulus time histogram. And you can see that the visual response here at peak is about 25 spikes per second, and there's a much bigger motor response. Now, let's compare that to block 2, block 1. So block 1 in red is shown here at the top, and the peak here is 3 times higher, 75 spikes per second. Now, this is a visual stimulus. How the heck does the number of expected drops change the firing rate of a visual neuron? You notice that it's not amplifying everything because the motor response here is exactly the same. So there's something special about the visual input to this neuron that has changed. And this is just now one of many, many, many examples where expectation modulates sensory responses. And this is, my interpretation of this is that the visual system is not in the business of representing a platonic visual representation of the kind that people in computer vision are looking for. No, it's representing not just what's out there, but the value that that object has for you. And if a particular object or task or response is more valuable, then you're going to represent it with greater fidelity or greater strength. And just to bring this home, in these 2 blocks, the sensory input is identical and the motor output is identical, right? So there's, in terms of behavior, in terms of the task, what you've got to do is identical. The only difference is something inside the brain which is modifying the responses. Okay, well that brings us one step closer to what I think is really going on. This is the biological input. Making decisions, okay. Suppose that you have this difficult decision to make. Are you going to go for the hot water or the cold water? Well, that depends on whether you're coming in from a cold winter here in Lethbridge, in which case you might want some hot tea, or if you're coming in after a run and you're sweating and you want something cold, right? So it's clearly your state, your behavioral state, whether you're thirsty or not, whether you're hungry or not, is going to affect your decision, and that decision could affect neurons in the sensory systems as well as the decision areas and the motor system. So this is really, I think, going to change this assumption about that you have these modules in the brain and that each one is dedicated to a particular task. I think there's something fundamentally flawed with that. So here's the conclusion of our essay. Obviously, visual systems evolved not for the achievement of sophisticated visual perception as an end in itself, but because visual perception can serve motor control, the final common pathway, and motor control can serve vision to better serve motor control, and so on. What evolution cares about is who survives, and that means basically who excels in the 4 F's: feeding, fleeing, fighting, and reproducing. How to exploit that evolutionary truism to develop a theoretical framework that is, as it were, motocentric rather than visuocentric, we only dimly perceive. So the conclusion we came to was that we got it backwards, that we really should be thinking about the visual system as a handmaiden to the motor system, to help the motor system do the right thing to survive. And the same thing is true of all the sensory systems. In fact, this got to be the case that that the motor system is actually the place where you can't do two actions at the same time. So therefore, you do have to make a decision of what you're going to do, reach out or not, or say something or not, right? So in a sense, it's really the final bottleneck. And it's an important one, because if you make the wrong decision, you make the wrong movement, as you'll see later in the talk, it could be toast. Everything changed in my life on April 2nd, 2013, when President Obama announced the BRAIN Initiative. I was in the audience. I was actually involved in bringing it to the White House. I was involved in the committee that wrote the report for NIH to set priorities for the BRAIN Initiative, and it's about halfway through now. And it has succeeded way beyond what I or the other 14 people on the committee that wrote the report had ever imagined. Our goal was to develop innovative neurotechnologies, not to cure disease, because there's already a huge amount of money going into that, and that's very important. But if we could develop tools and techniques that could accelerate the research, then everybody is going to benefit. It's like a tide that raises the level of all the boats. And in particular, one of our high priority goals, we had 7 of them, was to develop tools and techniques to be able to record from more neurons at the same time, from more different brain areas. And that more different brain areas has really turned out to be the really key step that has really changed my mind, and yours, I hope, in a minute, about how to think about behavior and how the brain is representing it. So here's the way that almost all experiments have been done up until recently, which is I'm interested in vision. I'm going to put my electrode into the visual cortex, and I'm going to present visual stimuli. And lo and behold, you get responses. Okay? But there's a million other experiments you could have done. You could have changed the amount of juice. Right? That was one of millions of experiments. So you have no idea what else that neuron might respond to, or under what conditions it might respond. Whether the animal is sated or not, or whether it's thirsty, may affect that neuron's firing too. But now, and this is within the last 5 years, it has become possible to record from tens of thousands of neurons simultaneously from dozens of parts of the brain. This is a paper that came out earlier this year from Ken Harris and Matteo Carandini's lab at UC London. Here they're using something called NeuroPixels. This is a new probe that has been developed. It's very long. It's about 1 centimeter long, so it can go through the entire brain of the mouse. Has about 1,000 contacts. And in this particular study, they had about 10 of these probes, so they could record 8 probes. So they can record on the order of 10,000 neurons. They also, in a separate series of experiments, use calcium imaging. That's another way you can record from thousands of neurons, is by having a genetically encoded calcium sensor. And when neurons are highly active, you get a lot of fluorescence. But here, the focus is on these NeuroPixel recordings, because you can get single spikes there much more easily than with the calcium probes, which is a secondary signal. So here's what they did. It was a head-fixed, awake mouse, just resting. There was no task. It's just sitting there. Now, have you ever looked at a mouse when it's just resting? Bruce? Okay. They're never at rest.
The brain is never at rest. The muscles are at rest.
The whiskers are whisking. Well, you have particularly laid-back mice, okay? No, the snout, the sniffing.
Talking about rest or sleep?
I'm talking about just quiet rest, when, not sleep. And they had a camera, and the camera was keeping track of eye blinks and squints and snout, sniffs, and the whole face has activity. And then what they did was to use a motion algorithm for detecting motion, the flow, and because they wanted to see differences. And then they applied PCA, principal component analysis, and they used the top 3 principal components. And you can see that positive is when things are moving in one direction, blue in the other direction. And then they compared the activity patterns of the neurons with the spontaneous motor behaviors through this preprocessing. And they asked the following question. If we know what the firing rates of the neurons are, can we predict what the actual movements of the face were? And by using linear regression, they were able to show that you can predict the actual movements with 80% of variance. And vice versa, if you know what the movement is, you can predict the activity patterns. Now, what's interesting here is that these electrodes are placed all over the brain from 40 different brain areas. There's information about what's going on behaviorally in every single area that they recorded from, including the thalamus, the superior colliculus, outside of the motor cortex, sensory cortex. And this is something that has been seen many, many times. Everybody who puts their electrode into visual cortex says, oh, there's spontaneous activity here, maintain activity. And of course, people ignore it. It's just the background, it's noise. It's not noise. It encodes the entire behavior, probably not just the motor behavior on the outside of your skeletal muscles, but probably your visceral system as well, which is also represented, we know, in different parts of the cortex. So this is really, I think, the smoking gun, and this is just the beginning. In the same issue of Science, there were 2 other papers showing that you can modulate all the activity in, this is Carl Dijsselroth's lab, the activity in neurons, 40,000 neurons throughout the entire brain of a mouse, depending on whether it was thirsty or not. Just think about that, right? This is a new era. We really have to rethink. We have to change. I think this is just like a Brenda Milner moment for neuroscience. We really have to rethink, some of our assumptions, and think about this is very important, by the way, for solving a problem that is a false problem, which is the unity of consciousness, right? I mean, people are saying, well, if you have perception here and you have decisions here, how do they form some kind of a sense that we have a uniform, integrated view of the world through all the senses and the motor systems? And again, this makes it less of a mystery if it's the case that every single neuron in the brain, or the majority, I can't say every, but at least from the sample, a majority of the neurons in the brain have access to all of those decisions that are being made in every single muscle and every single part of your body. So this is a starting point for this talk, which is going to be focused on the motor system. And this is a project that has been going on in my lab in collaboration with John Doyle at Caltech, who's a control theorist. And our goal was to try to understand something about what we call speed accuracy trade-offs. This is something that we have to understand biologically, but it turns out that this is a really big problem also in control theory. And here's the problem. The problem is that I want to be able to reach rapidly, but also accurately. Psychophysics is very well studied, and I'm going to show you the data. But the problem is that you're dealing with a nervous system that has nerves that are very, very slow by standards of control theory. Tens of milliseconds of delay, which for a negative feedback loop causes all sorts of problems if you want to be accurate. Deals with limited data rates. In a computer you can have gigabits per second going to your computer, through Ethernet, but in brains we're talking about bits per second, on even the big nerve. So the biology has to deal with these components that are really not necessarily very fast or accurate. And that's going to be our attempt to understand that, find some principles. Now, this is a diagram that sort of illustrates all the different levels that neuroscientists look at the brain, from the molecular to the entire central nervous system, and focusing on things that, in the last few minutes, I've been focusing on things down here, and we want to link them to things up here. So we're trying to make a link between systems level behavior and the properties of the actual mechanisms underlying that behavior. And I'm going to use something that's very familiar to all of you, I hope, just to illustrate how a control theorist thinks about the brain. Okay, this is a simplified diagram of the ocular motor system that's responsible for smooth eye movements. Okay, so I want you first to do an experiment. I want you to put your hand in front of your face and move your head back and forth, and move it very fast. You can still see the fingers, right? Even though you're moving your head very fast, 5 hertz or something. You can go up to 10 hertz, 15 hertz. Okay. Now, what I want you to do is the same experiment. Put your hand in front of your face and move your fingers back and forth. Keep your head still. Okay. It turns out that if you go above 1 or 2 hertz, you can't see it clearly anymore, right? There's a discrepancy here between moving your head and moving your hand. Why is that? Well, the reason is that there are actually 2 different systems for guiding your eye when your body is moving. There is a vestibular ocular reflex that detects head motion from semicircular canals. So these are vestibular inputs. And they have a very fast pathway. It's on the order of 5 to 10 milliseconds to the eye muscles. And it's a feed forward. And that's why you're able to stabilize the image on the retina even though your head is rotating very quickly. And that's very important because otherwise you're going to be confused as you walk around because your head is bouncing around, right? But there's a slower pathway which comes from the eye, and that's the image on the retina, which is processed by the visual cortex, and it will eventually lead down to the same actuator, so you can track a slowly moving object. It's called smooth pursuit. But there's a 100 millisecond time delay there. And that 100 millisecond time delay basically makes control very, very difficult. However, when I'm bouncing around as I'm walking, I can track a slowly moving object because I have a layered system which on the bottom has very, very fast but not necessarily accurate way of being able to stabilize the image on the retina. As long as it's within a few degrees per second, it's not a problem. And a slower system that is able to handle things that are on a slower time scale and be able to accurately track and recognize license plates and things like that. So this is really, the concept here is that you don't do it with one system, you do it with multiple motor systems that are layered, that together perform better than either one of them alone. Okay, so here's the data from speed accuracy trade-off for reaching. It's called Fitts law. It's been replicated thousands of times, and it's true not just for reaching, but it's true for saccading to a target. It's true for using your mouse on your computer. And here's the observation, is that if you have to reach a distance d to a target that has a width w, that's the accuracy, then the time it takes can be accurately fit with this equation, with 2 terms. One is a constant, and the other one is a log of a ratio of the distance to the width. And what this means, though, because log is a very slowly increasing function, that you actually have a fairly high degree of accuracy, even if you're going a long distance. And so this is really what we're going to try to understand. I mean, how is it that the brain is able to accomplish that? So here's one of the secrets that the brain has that everybody knows about, but it's going to play a very important role in explaining Fitts law. So let's look at the different nerves. We have a big sciatic nerve that goes several meters down to your tip of your toe. It's fast, but it's inaccurate. It's because it's big, it takes up a lot of space. So there are only relatively few of them. On the other extreme, the olfactory nerve has 100 million fibers, but they're tiny, micron size. And that makes them very slow, very, very slow. But maybe you can give up on speed when you're smelling things, because things don't change that fast when you're smelling things. However, because of the fact that you have 100 million, you have an enormous number of bits going up into the olfactory, which is good because olfactory space is very large. There's an unbelievable number of different odors that could exist, and so therefore you have to have a very high degree of high dimensional representation of the odors. So it's just the right thing to do. It's slow but accurate, and everything in between, right? I call this, John and I and our students who are working on this project, the diversity sweet spot, because you're going to see that having diversity, having some fibers that are fast and inaccurate and others in between, all the way to slow and accurate, will allow you to be able to have the best of both worlds. Okay, so this is the only equation, and I just want to go through it just to, this is a fundamental relationship between the data rate and the signal delay for an axon. And I'm not sure if I should go through the derivation except to say that what's important here is you have a certain cross-sectional area, and you can use it either for one big axon that's fast, or you could use it for a bunch of small ones that are slower. But it turns out that regardless of what the caliber is, whether it's a big one or a small one, they all have this relationship that the data rate, this bits per second, is proportional, this is a constant, to the time delay. And it turns out from that, you can actually work out the control law underlying Fitts law. So here's a control theory diagram which basically summarizes what the reaching system has to do. There is somewhere, and this is a way engineers think about this, not the way biologists do, but you'll see that we can map this onto different parts of the brain. So at some point, you have a controller that's going to tell you to move your arm a certain distance. That issues a control, which is a signal, which is a function of time, to the muscles, which then reach out into the and try to achieve that accuracy. Of course, if there's an error, the visual system will pick that up, and this is the slow pathway. This is a faster pathway. And then there's some noise and uncertainties. And now here's the problem, is that in these pathways, there's a limitation on the rate, and there are time delays, and the visual system has this really long time delay in terms of the 100 millisecond processing time. P, if you're interested what P means, it's plant. And this is not a plant that grows in the ground. This is a processing plant, because engineers control how material flows through the chemical processing, and the plant can also be a robot arm. But here's that comes from John's lab, which is the control theory that this delay has two components. One of them that is linear to the time delay in this system, internal time delay, and another one which is inversely related to the data rate and is proportional to the log of the distance traveled divided by the accuracy. This should be familiar. This is Fitts' law. And what this is telling us is that the constant in Fitts' law is telling us something about the internal delay, and the constant in front of this log relationship, which it picked up, is telling us something about the limitation on the rate at which information is transferred within the brain. Okay. Now, what makes this unusual for a control theorist is this R. Because when control theorists build systems, they don't use, low bandwidth wires. They use gigahertz wires. And so for a control theorist, they could almost completely ignore the R when they create a controller. They only worry about time delays. Okay, so that's interesting. So biology does have to worry about the data rates. But now here's the flip side of the coin. So control theory is one part of engineering. Information theory is another part of engineering, right? And they go to separate meetings. You have very little in common. And if you ask an information theorist, he'll say that, oh, it's all about data rates. It's all about, how much information you can get through a channel. It's all about coding. It's all about information transfer. But they never consider time delays. They assume that, well, it's going to happen, and it doesn't matter how long it takes to get there. I just want to know I just need to get the information to the cell phone, right? So they completely ignore time delays. So this is interesting because nature has to worry about both together, whereas engineers have split it down the middle, and one worries about T and one worries about R. Interesting. So John and his students put together a game which, as you can see, is based on a little driving input. This is a controller which you can steer. And here's what the subject sees when they're playing the game. The idea is to collect data to try to see whether or not this theory that we have is actually can be confirmed experimentally. And by the way, this is a kind of a game that nobody, as far as I know, in psychophysics has ever looked at before. But you'll see that it has actually a very interesting outcomes. Okay, so here, you start here. This cursor here is where you're controlling. And your goal is to get it to the middle of this target area. And we can vary W, we can vary D. And we could collect data. Now, in addition, because of the fact we control the interfaces, we can add delay, for example, to either the visual display or to the motor display. So just put a delay in. We can also vary the data rate by quantizing the angle of the steering wheel. So, one bit would be either right or left. And if you had 2 bits, now you can grade it more finely, fast and slow, right? And so we could vary that, data rate and delay. And this is what theory tells us. Theory tells us that as you, and this is now the minimum reaching time, if you add extra delay, remember the delay was the T, was a constant. So you increase the constant, you're increasing this TR, and that goes up linearly. And remember, the rate was 1 over R. In fact, here it's right here. So as you increase the data, as you reduce the data rate, as you increase the data rate, the accuracy will get better and better and better as 1 over R. So this is theory. Here's experiments averaged over about 6 subjects. You can see the standard deviation surrounds the mean. And so here's the delay error, which is pretty linear. And here's the rate error, which comes down approximately hyperbolically. And here's the total error. We just add these two together. And we approximate what we expect here. But this is actually making a huge assumption. We made a huge assumption that these two types of error are additive. It could be nonlinear. It could be that they interact with each other. And the only way that they could be adding linearly is if in some ways the two systems are independent of each other, so that no crosstalk. And that's a prediction. So that's one illustration of the diversity sweet spot, but it turns out there's an equally interesting diversity sweet spot on the motor side. So first of all, we know that there are 2 different types of muscles. There's a slow twitch and a fast twitch. The fast twitch is the fast twitch is very fast. I mean, you use it for explosive activity, but it's inaccurate. It gets you off the ground, but it doesn't really take you very far. Whereas the slow twitch are very accurate, but you can't get, you're using it slowly and you can go long distances, but it's not making very fast, rapid movements. Yes?
Sorry about the delay in my question. So with those two systems, are you thinking that people are using one or the other?
No, no, no. No. If you've ever eaten turkey, right, there's dark meat and there's light meat. That's these two different types of mussels. And mussels often have both.
Sorry, I meant the steering wheel.
Oh, you were talking about the steering wheel.
The total error. So are people using one system or the other? Or are they somehow blending?
No, they have to use both. That's to say they're using their visual system to figure out where the target is, but then they have to use the FAST system in order to be able to get to the, launch it properly and get to the middle of the target. Yeah, so and the control theory for those two different systems is part of the way that the control theory is set up. In any case, not only that, but there's something called Hennemann's size principle. And this has been known now for over 50 years that in the spinal cord there are motor neurons that are recruited sequentially. First, the very fine muscles with low force, and then as you are increasing the force, you will recruit higher and higher strength muscle fibers. So that's another example of diversity sweet spot where you are using a range of motor neurons, not just a single force, in order to be able to control your limbs. And so we put in the diversity sweet spot not for the axons, but for the motor fibers. And here's what the theory says, that as we change the width over D, if it's uniform, if you only have one strength of muscle fiber, your reaching time is much, much longer than if you have a diverse set of muscles. And here's now where we ask the subject to either go at a uniform rate or to be able to vary their strength fast at first and slow later. So you just say, just go at a constant rate to get to the target. So diverse is good because you can explode off the start line, and then you could, as you slow down to get to the target, you could be more accurate. And here now is the kind of the key, which shows us that we're able to explain the Fitz's law. Remember, Fitz's law only depends on this ratio of D to W, distance to width. And this is 4 different distances, but you see the width increases linearly so that they have exactly the same ratio. And according to Fitz's law, they should have exactly the same reaching time. And so here we are. Uniform goes up linearly because the farther you're away, you're going at a constant rate, so you can't go too fast. And uniform fast works good for things that are close, but not so fast. When you get to a distant target, you're way off, and so it's going to take longer to get to the target. And if you do both, if you allow fast and slow, normal, you can see that Fitz's law is correct. It's independent of that. It's a function only of the ratio. Okay, now I want to take what I've done with Fitz's law and generalize it to mountain biking. So how many people here mountain bike? I see Bruce is not putting his, I know that he's a glider pilot, but he doesn't mountain bike. Okay, he's smart. Okay, so I'm going to play a video from YouTube, and I want to warn you that if you have acrophobia, fear of heights, you should not watch this. This is insane, what you're going to see. This is nothing until you'll see it toward the end. It gets totally insane. mean, he's inches away from a sheer cliff.
How's the wind? Oh. I don't like the wind.
Here we go.
Whoa.
Oh, Jesus. Whoa. It is. These are the successful ones. He had a GoPro camera. Okay, so this is John Doyle's picture of what you're seeing here. Is that just like we had for Fitts' law? There's a trade-off here. If you have to have really good reflexes because you're bouncing around and you have all kinds of reflexes to keep the bikes steady, keep it from going over. But you also have to plan. You have to see, when is the jump going to occur, and should I move it to the right or left, and so forth. And so, so the question is, how do you manage to put together these two very different systems and make it appear as if the rider is actually here, fast and accurate. In order to survive, you have to be both fast and accurate, right? But there's no single system that does that for you. So here's now the control system. We've put in there's internal delays, and the key thing here is that there's warning 100 milliseconds ahead that you can plan. And I'm going to go through this very quickly because it's very similar. The game now is similar to the previous one, except that your goal is to follow, this green cursor should follow this path, which is going to zigzag. And as you can see, you have some warning, and we can vary the amount of warning, how far into the future you can see. For example, if the warning was just an inch, you wouldn't be able to see this coming and prepare for it. We can also put in extra time delays. We can put in quantization errors. And here's the data. This is the error, and here's the bumps only. You can see every bump is put into the right and the left, and it's corrected. And if you do the trails, you can see that there's an error at the turn, but more or less stays on track. So this is really, you can collect lots and lots of data very, very quickly. It's a very nice setup. Here's two subjects where we're changing either the time delay or the data rate, and we get, as we saw with Fitts, they follow these curves to the upper right, lower right, and now compare the sum, which is t plus q, dashed line, to the sum of these two lines, the red and the blue, which is the dark black. Changing both. So you're doing both at the same time. The subject is following it. Is pretty much additive for both subjects. And this tells you what the range of parameters were. So this is amazing. So this is telling us that trail following is similar to Fitts in the sense that you can do both. You can see ahead. You can plan, and they seem to be independent of each other. And so here's now some raw data. And what we did was we looked at the error for each subject is a dot. And you can see the errors are smaller for the bumps and the trails. And then when we had them do both, the errors were larger. But in fact, if you sum these two, it is not significantly different. So it's nice to see data like this because it really means that we're dealing with a system that probably is independent. So there's a delayed system here. This is the one that has the very fast feedback loop, and this is the one that has the slow planning. But they feed into the same output. Okay, now here's my message. Okay, I've given you two examples. both for motor control, but the actual concept here is that with diversity, you can make things appear on the outside as if you have the best of both worlds. So diversity is good, and biology exploits it, both on the motor side and on the sensory side. But here's an example from a completely different industry, transportation. Okay. So suppose that this is now time to reach your destination, and you can get there many different ways. But let's just say that if you walked to your destination, it would take 200 hours. It would take you a couple of weeks, and if you like walking, that's great. But if you're like me, 200 miles is just about the right size from LA to San Diego. Well, I'll take the plane. And the plane really knocks that down by 2 orders of magnitude, right? So now it's going to take maybe a couple hours if you count going to and from the airport. But the airplane doesn't have much accuracy. It's not going to take you to your home, right? It may take you 25 miles away, right? The accuracy isn't very, it's fast, but not accurate. Just like your sciatic nerve. But you can take the bus, you can drive, and that's pretty good over tens of miles, right? So it takes you an hour to drive. And then finally, once you get close to your home, you can walk that last mile. That's only going to take you 15 minutes. And if you stretch these together, you can get from LA to San Diego all together with these 3 different diversity mechanisms of transportation in about the same time that it would be if you just flew all the way point to point. Maybe a little bit slower, but you can see it's only going to take a few hours. So this is the way our transportation works. I mean, in fact, if you look at any engineered system that is efficient, it's not just a single method for solving it. Multiple methods, and you use them sequentially or in combinations. And I think this is a general principle that biology has really exploited in many, many different ways. And by the way, it's also very energy efficient. If you had to load yourself up with squid giant axons for everything, you'd explode because of the energy requirements. It's much better to use the mechanisms that are the most efficient. Okay, so I want to thank everybody for their attention, but really wanted to have a shout out for John and Yori and Kuan Ying, who are responsible for the driving experiments, and we're really also responsible for thinking through some of the deep engineering issues. And this is actually a very interesting collaboration that we have, because from John's perspective, this is illuminating control theory in a way that most control engineers would never ever think of, because of the fact that we're dealing with different constraints in biology. But these are constraints which actually turn out to be important in something called distributed control systems. Something that control engineers cannot do now is to instead of having a single executive, a single place where decisions are made, and then issuing all the commands for the control from one place, gathering all the sensory information and making decisions. If you look at biology, right, we have layers of controllers, we have distributed sensory information coming in at different levels. And there is no theory for that in engineering. But biology obviously has figured it out. So the real, I think, potential here is to reverse engineer distributed control in animals and take that into engineering in a way that would really change the way that engineers think about their problems. So thanks again for your attention, and happy to take any question. Bruce.
Lovely. So, coming to the mountain bikers, some survive, and some don't. The ones who survive, some get there first, and others a little more slowly. what is it that makes the difference between the winner and the loser of those survivors? It has something to do with training of the planning system and learning about what features to be planning with. And so that should be a central part of the theory here, is what goes into efficient feature recognition and the use of those features.
Yeah, so Bruce is bringing up another issue, important issue, having to do with learning. To become good at anything, it takes a lot of practice, right? And by the way, I didn't mention it, but for this driving game, we let them practice for a few hours before we start taking the data, because of course they get better and better. They asymptote.
But in that test, there are no features, are there, that can give them better predictive value if they've learned about what features to recognize?
Well, they get better. I don't know why. I mean, I think that with any system, okay. So there's a lot of evidence what I'm about to tell you, but not for these particular games I've showed you. It looks like what is happening is that at the beginning of learning how to play any tennis or any other game, that you're using your top-level controllers in order to get into an initial state where your body is in the right position, and that takes conscious effort. But with practice, that's downloaded to a lower layer so that you don't have to think about it anymore. Your body automatically goes into the right configuration, and the lower level controller could do more of the work. So what you're doing is you're pushing it down. And people who are super at a sport like basketball have pushed it so far down that they can do amazing things like that mountain biker, right? That mountain biker's not thinking his feet or his arms anymore. He's in the groove. He's basically really focused on what the next maneuver is going to be, and he's kind of planning ahead a little bit. But even that probably is pre-planned in a sense. He's done things like that thousands of times, so he knows what he has to do. And sometimes he waggles his tail. You notice that he was showing off there. So the learning part of the story is a fascinating one, which we haven't even. It's called adaptive control. It's a whole branch of engineering of control theory, and it's clearly going to be the next step. In fact, John was telling me that one of his former postdocs, actually, who's at Berkeley now, has been bringing some of the concepts from control theory into machine learning. And the idea would be to really to combine the two, and to be very powerful for being able to not just program as a controller, but to be able to adapt it to new conditions, to generalize. Another question.
The issue of mass action versus localization of function, probably the oldest problem in neuroscience. When you presented Harris' experiment, you really took a strong position, it seems to me, for mass action. But you could interpret each of those neurons as having a localized function, couldn't you?
Well, thank you for asking the question. The question is, if you go back to the early days, and this is Lashley, I think, did these experiments where he basically scooped out parts of the rat brain and showed that the more he scooped out, the worse it did. He interpreted that as mass action. But, and subsequently, there's been a tremendous amount of data that has come in showing that specific parts of the brain seem to be specialized. And they're both, I think they're both right. So I didn't go into the details of this, the Harris experiment. But what they showed was that if you, in addition, simply have the rat sitting there, you give it a stimulus, like a visual stimulus, that the visual response is superimposed in the visual parts of the cortex on top of this ongoing background behavior. So therefore, and they said it was orthogonal, which I don't believe. That is to say that the separate neurons or separate ensembles were being activated by the sensory stimulus and all the behavioral signals. So my guess is that just like you may want to modify your perceptual representation by the state of your body, whether you're thirsty or you're hungry, or whether you are in stress and so forth, you might also want to be able to vary it according to whether your body is in the right posture or not to detect the stimulus, whether or not you're afraid and you're tensing, right? You want the sensory system to know that instantly. You don't want to have to wait to propagate it from one area here to another area here. So I think that the behavioral state is just very, very useful information everywhere. Everybody should have access to in case you need it in order to make decisions. And by the way, this is another experiment that came out from that lab. So they had another a case where the animal had to do a task. It had to press a lever. I think it was a olfactory discrimination task. But they were recording now from 40 areas. And the question is, where is the first signal that is correlated with the decision? And it wasn't in the cortex, or everybody who studies the cortex thinks it's in the cortex. It's not. It's in the first signals are from the reticular formation in the brainstem, and the superior colliculus, deep layers of the superior colliculus. Again, this is not something anybody would have predicted from looking at the textbooks. Everybody thinks, oh, decisions are made way up at the top there and they're propagated down. No, in this case here, for an animal, that it really is well practiced. It's able to make those decisions at a much lower level. In fact, it's related to your question about if you're really, really good at something, you don't need to have the cortex engaged on a moment-by-moment basis. And so, the beauty is that the sensory representation is there too in case you need it. In other words, you have a motor, kind of a field, mean field, not mean field, but a broadly available motor. In fact, okay, let's be specific. Where is it coming from? Where are those signals coming from? Well, we know that the cerebellum, which receives proprioceptive information from almost every muscle in the body and is corollary discharge, right? Efferent's copy from the motor signals. So it knows what the motor system is trying to do, and at the same time it knows what's coming back from the periphery. All that information gets into the cerebellum, and that projects back to the cortex, right? So there's a tremendous amount of information that's going from the bottom up to the cortex, which could support all of that behavioral information. And why it's there, I gave you some speculation, but it seems to me now that we know spontaneous activity actually isn't noise, we should be studying it. We should actually say, hey, let's focus on what that signal is and how it interacts with the sensory information coming in. So that's that's for the future.
To continue that question, does that mean that this spontaneous information is reflections of information generated by specialized areas like the cerebellum collecting it?
Yeah, that was just my speculation. I mean, I know the cerebellum, it receives information from many areas and sensory feedback, and it projects even it's really interesting, the lateral cerebellum projects actually the prefrontal cortex. Everybody thinks it's a motor structure, but if it's projecting prefrontally, that means it's part of the planning system too. It's not just a sort of a coprocessor that keeps track of what's going on at a lower level. But there are many, many other sources of information that the cortex has access to. There's neuromodulators, for example, that are very important, that are regulating gain of many circuits. So it's just that I think we've gone too far in the localization business in terms of focusing on just one type of response a neuron can have to a sensory stimulus at the exclusion of all the other inputs it's getting. And just to give you another sense for anatomical, I mean, again, these facts are in the literature. Everybody knows them, but nobody has put them all together. And this is another thing from our chapter. Okay, so it turns out that if you look at the LGN inputs to V1, right, the number of synapses in the cortex in V1, for every thalamic input, there's 10,000 inputs from other parts of the cortex and local circuits and from other feedback connections from V2. So the vast majority of all of the signals that the neurons are getting is not coming from the outside, it's coming from other parts of the brain, right? And even on, the ones that are focused on layer 4 spiny stellate cells, only 10% of the synapses on those are the ones that are being driven. Only 10% are coming from the thalamus. So that immediately tells you that the modulation that is determining how responsive the cell is and the cortex is dominant. That's dominant. We can't ignore it. Yes, it's true that you can get visual input in, but that's not the only thing that's getting in.
It's all coming from all different areas of the brain getting integrated together.
Exactly. That's the take-home message. If you don't remember anything else, it's just we have to take seriously all of the collaterals and all of the other parts of the brain that are equally important. And also, I think this reflects on Bruce's advice to the young investigator, which is that it's all about collaborations. And so the vision people should be collaborating with the motor people. By the way, that's not the way it is now. It's really interesting. When this came out, this book chapter, the vision people hated it. The motor people loved it because they knew all along that the motor part was the most important part of the brain. But nonetheless, I mean, that's the point, is that we're now in a position where we really have to think about teams who have different perspectives, who have different tools and techniques working closely together.
This is more of a comment than a question, but interesting to see what you think. What you were just saying about collaboration and vision versus motor. Your chapter, it's been a while since I read it, 94, was already a couple decades after Gibson. And
We had a figure from Gibson.
right, and so
Yeah, affordances.
it had already gone through this cycle where you had Gibson and then you had Marr. And you also had this transition from behaviorism to a cognitive revolution. And this idea that Marr was floating on, essentially, that we had these internal maps and these internal representations. And that has led to things like modularity, and that you would have this particular image inside the brain and how you get and so on. But the comment really is that one thing that I like very much about your work is a return to this idea of the perceptual motor integration, the evolution, the ecological psychology of it, that we are living, that, for example, we have more descending fibers from neuron down to LGN and vice versa.
By a factor of 10.
Yeah, and by a factor of 10, we ignore all of that. And in that some of these would have been, some of that would have been.
Oh, it's getting late. It's getting late. Could you shorten your comment?
And the idea basically been that some of this approach,
Right.
the modularity as opposed to parallel distributed systems, the idea of cognitive models.
Right. Okay, so, this is a fantastic topic that I would love to talk about. Let me just make the comment that one slide I showed you from Paul Glimcher, together with an incredible breakthrough that was made back in the 90s by Wolfram Schultz with dopamine neurons, and Peter Dayan and Reed Montague, who are postdocs in my lab, showing that these dopamine cells were doing reward prediction error, has given rise to a huge shift in the way we think about, it's called the neuroscience of economics, neuroeconomics. In other words, realizing that human beings aren't just perfect value optimizers, but rather are there because of the fact they have biology driving their decisions. So there's a whole story there that you're right is shifting. And interestingly, it actually goes back to the behaviorists, right? The behaviorists were into that kind of a system, reward-based learning systems. So, yeah, I think this is we're living in an era now, which is, I think, going to be, 100 years from now is going to be seen as the golden era of neuroscience, because we have the tools and techniques to actually answer questions now. Whereas before, the best we can do was, a little here, a little there, and wasn't really answering the questions. But I think we will. Within the next generation or two, I think we're going to really get to the point where we will have a theory of the brain.